Dear friends, I am Dr. V.K. Dikshit, retired professor of Pharmacy, Department of Pharmaceutical Sciences, Dr. Hari Singh Ghosh, Central University. We are discussing WHO guidelines on quality control of herbal medicinal materials. In this lecture, we will be dividing it into 5 modules. Module 1, determination of hemolytic activity. Module 2, determination of foaming index. Module 3, determination of tannins. Module 4, determination of swelling index. And Module 5, determination of microorganisms. Module 1, determination of hemolytic activity. Many herbal materials, especially those derived from the families Caryophyllaceae, Araliaceae, Sapindaceae, Primulaceae, and Dioscoreae, contain saponins. The most characteristic property of saponin is their ability to cause hemolysis. When added to a suspension of blood, saponins produce changes in the erythrocyte membranes, causing hemoglobin to diffuse into the surrounding medium. The hemolytic activity of herbal materials or a preparation containing saponins is determined by comparison with that of a reference material saponin R, which has hemolytic activity of 1000 units per gram. A suspension of erythrocytes is mixed with equal volumes of a serial dilution of the herbal material extract. The lowest concentration to effect complete hemolysis is determined after allowing the mixture to stand for a given period of time. A similar test is carried out simultaneously with saponin R. Procedures proposed for determination of hemolytic activity of saponation herbal material are all based on the same principle, although the details may vary. For example, the source of erythrocytes, methods for the preparation of erythrocyte suspension, and the herbal material extract, the defined hemolytic activity of the reference material of saponin, and the experimental method. In order to obtain reliable results, it is essential to standardize the experimental conditions and especially to determine the hemolytic activity by comparison with that of saponin R. Recommended procedure: to prepare the erythrocyte suspension, a glass stoppered flask is filled to 1/10 of its volume with sodium citrate 36.5 gram per liter test solution, filling to ensure that the inside of the flask is thoroughly moistened. A sufficient volume of blood freshly collected from a healthy ox is introduced and shaken well immediately. Citrated blood prepared in this way can be stored for about 8 days at 2 to 4 degrees centigrade. Place 1 ml of citrated blood in a 50 ml volumetric flask with phosphate buffer pH 7.4 test solution and carefully dilute to volume. This diluted blood suspension, that is 2% solution, can be used as long as the saponate fluid remains clear and colorless. It must be stored at a cool temperature. To prepare the reference solution, transfer about 10 mg of saponin R, accurately weighed, to a volumetric flask and add sufficient phosphate buffer pH 7.4 test solution to make 100 ml. This solution should be freshly prepared. The extract of plant material and dilution should be prepared as specified in the test procedure for the plant material concerned using phosphate buffer pH 7.4 test solution. Preliminary test. Prepare a serial dilution of the plant material extract with phosphate buffer pH 7.4 TS and blood suspension 2% using 4 test tubes. As soon as the tubes have been prepared, gently invert them to mix, avoiding the formation of foam. Shake again after a 30-minute interval and allow to stand for 6 hours at room temperature. Examine the tube and record the dilution at which total hemolysis has occurred, indicated by a clear red solution without any deposit of erythrocytes. Proceed as follows. If total hemolysis is observed only in tube number 4, use the original plant material extract directly for the main test. If the total hemolysis is observed in tube 3 and 4, prepare a 2-fold dilution of the original plant material extract with phosphate buffer pH 7.4 test solution. If total hemolysis is observed in tubes 2 and 3 and 4, prepare a 5-fold dilution of the original plant material extract with phosphate buffer pH 7.4 test solution. If after 6 hours all 4 tubes contain a clear red solution, prepare a 10-fold dilution of the original plant material extract with phosphate buffer pH 7.4 test solution and carry out the preliminary test again as described above. If total hemolysis is not observed in any of the tubes, repeat the preliminary test using a more concentrated plant material extract. Main test. Prepare a serial dilution of the plant material extract, undiluted or diluted as determined by the preliminary test with phosphate buffer pH 7.4 test solution and blood suspension 2% using 13 test tubes as shown in table 4. Carry out the dilutions and evaluations as in the preliminary test but observe the results after 24 hours. Calculate the amount of medicinal plant material in gram or of the preparation in gram or milliliter that produces total hemolysis. To eliminate the effect of individual variations in resistance of the erythrocyte suspension to saponin solutions, prepare a series of dilutions of saponin R in the same manner as described above for the plant material extract. Calculate the quantity of saponin R in grams that produces total hemolysis. Module 2. Determination of foaming index. Many medicinal plant materials containing saponins can cause a persistent foam when an aqueous decoction is shaken. A considerable number of plants have saponin as active, commercially important constituent. Ginseng, Dioscorea, licorice, Brahmi, Karela, etc. are examples of saponins containing drugs. The foaming ability of an aqueous decoction of plant material and their extract is measured in terms of foaming index. Recommended procedure. Reduce about 1 gram of the plant material to a coarse powder, sieve size number 1250. Weigh accurately and transfer to a 500 ml conical flask containing 100 ml of boiling water. Maintain at moderate boiling for 30 minutes. Cool and filter into a 100 ml volumetric flask and sufficient water through the filter to dilute to volume. Pour the decoction into 10 stopper test tubes, height 16 cm, diameter 16 mm, in successive portions of 1 ml, 2 ml, 3 ml, etc. up to 10 ml, and adjust the volume of the liquid in each tube with water to 10 ml. Stopper the tubes and shake them in a lengthwise motion for 15 seconds, 2 shakes per second. Allow to stand for 15 minutes and measure the height of the foam. The results are assessed as follows. If the height of the foam in every tube is less than 1 centimeter, the foaming index is less than 100. If height of foam of 1 centimeter is measured in any tube, the volume of the plant material decoction in this tube is used to determine the index. If this tube is the first or second tube in a series, prepare an intermediate dilution in a similar manner to obtain a more precise result. If the height of the foam is more than 1 centimeter in every tube, the foaming index is over 1000. In this case, repeat the determination using a new series of dilutions of decoction in order to obtain a result. Module 3, Determination of Tannins. Tannins or tannic substances are substances capable of turning animal hide into leather by binding proteins to water-insoluble substances that are resistant to proteolytic enzymes. This process, when applied to living tissue, is known as an astringent action and is the reason for the therapeutic application of tannins. Chemically, tannins are complex substances. They usually occur as mixtures of polyphenols that are difficult to separate and crystallize. They are easily oxidized and polymerized in solution. If this happens, they lose much of their astringent effect and are therefore of little therapeutic value. Recommended procedure: prepare the herbal material extract, introduce the quantity specified in the test procedure for the herbal material concerned, previously powdered to a known fineness and weighed accurately into a conical flask. Add 150 ml of water and heat over a boiling water bath for 30 minutes. Cool. Transfer the mixture to a 250 ml volumetric flask and dilute to volume with water. Allow the solid material to settle and filter the liquid through a filter paper diameter 12 cm, discarding the first 50 ml of the filtrate. To determine the total amount of the material that is extractable into water, evaporate 50 ml of the plant material extract to dryness. Dry the residue in an oven at 105 degrees centigrade for 4 hours and weigh. This will be T1. To determine the amount of herbal material not bound to hide powder that is extractable into water, take 80 ml of the herbal material extract, add 6 gram of hide powder R and shake well for 60 minutes. Filter and evaporate 50 ml of the clear filtrate to dryness. Dry the residue in an oven at 105 degrees centigrade and weigh T2. To determine the solubility of hide powder, take 6 gram of hide powder R, add 80 ml of water and shake well for 60 minutes. Filter and evaporate 50 ml of the clear filtrate to dryness. Dry the residue in an oven at 105 degrees centigrade and weigh. Calculate the quantity of tannins as a percentage using the following formula: T1 minus T2 minus T0, the whole multiplied by 500 divided by W, where W is the weight of herbal material in grams. Module 4. Determination of swelling index. Many herbal materials are of a specific therapeutic or pharmaceutical utility because of their swelling properties, especially gums and those containing an appreciable amount of mucilage, pectin, or hemicellulose. The swelling index is the volume in milliliter taken up by the swelling of 1 gram of herbal material under specified conditions. Its determination is based on the addition of water or a swelling agent as specified in the test procedure for each individual herbal material, either whole, cut, or pulverized. Using a glass stoppered measuring cylinder, the material is shaken repeatedly for 1 hour and then allowed to stand for a required period of time. The volume of the mixture in milliliter is then read. The mixing of the whole herbal material with the swelling agent is easy to achieve, but cut or pulverized material requires vigorous shaking at specified intervals to ensure even distribution of the material in the swelling agent. Recommended procedure: carry out simultaneously no fewer than 3 determinations for any given material. Introduce the specified quantity of the herbal material concerned, previously reduced to the required fineness, and accurately weighed into a 25 milliliter glass stoppered measuring cylinder. The internal diameter of the cylinder should be about 16 millimeter, the length of the graduated portion about 125 millimeter, marked in 0.2 ml divisions from 0 to 25 ml in an upward direction. Unless otherwise indicated in the test procedure, add 25 ml of water and shake the mixture thoroughly every 10 minutes for 1 hour. Allow to stand for 3 hours at room temperature or as specified. Measure the volume in milliliter occupied by the herbal material, including any sticky mucilage. Calculate the mean value of the individual determinations related to 1 gram of the herbal material. Module 5: Determination of microorganisms. Medicinal plant materials normally carry a great number of bacteria and molds, often originating in soil, while a large range of bacteria and fungi form the naturally occurring microflora of herbs. Aerobic space forming bacteria frequently predominate. Current practices of harvesting, handling, and production may cause additional contamination and microbial growth. The determination of Escherichia coli and molds may indicate the quality of production and harvesting practices. Methods for determination are restricted. For example, the use of ethylene oxide has been forbidden within countries of the European Union. Treatment with ionizing irradiation is also forbidden or requires a special registration procedure in some countries. In addition, the presence of aflatoxin in plant material can be hazardous to health if absorbed even in very small amounts. They should therefore be determined after using a suitable cleanup procedure. Microbial contamination limits in medicinal plant materials. Different limits are set according to use of the herbal materials and the material itself. For contamination of crude plant material intended for further processing, including additional decompensed by a physical or chemical process. The limits adopted from the provisional guidelines established by an international consultative group are given for untreated plant material harvested under acceptable hygiene conditions. Escherichia coli maximum 10 raised to power 4 per gram. Mold propagules maximum 10 raised to power 5 per gram. For plant materials that have been pre-treated, for example with boiling water, are used for herbal teas and infusion, or that are used as topical dosage forms. Aerobic bacteria maximum 10 raised to power 7 per gram. Yeast and molds 10 raised to power 4 per gram. Escherichia coli maximum 10 raised to power 2 per gram. Other enterobacteria maximum 10 raised to power 4 per gram. Salmonella none. For other plant material for internal use, aerobic bacteria maximum 10 raised to power 5 per gram. Yeast and molds maximum 10 raised to power 3 per gram. Escherichia coli maximum 10 per gram. Other enterobacteria maximum 10 raised to power 3 per gram. Salmonella none. Detailed test procedures, culture media, specifications, methods of counting, strains to be employed, and methods of validation are described in WHO guidelines as well as in pharmacopoeia. Test for aflatoxin. This test is designed to detect the possible presence of aflatoxin B1, B2, G1, and G2, which are highly dangerous contaminants in any material of plant origin. Aflatoxin are one of the main reasons for rejection of supplies to Western countries. Aflatoxin are tested by thin layer chromatography test. The mixture of standard aflatoxin exhibit 4 fluorescent blue spots, and the hue and intensity of any of them should not match with the test material processed as per a specified method. Toxic residues. These may arise in fruit drugs as a result of pesticide application during cultivation and at a later stage from fumigation of stored products. The collections of herbal material from the areas where industrial waste is disposed could also be a source for toxic residues. The heavy metals and the insecticide and pesticide limits are a matter of great health concern. Thin layer chromatograph and gas chromatographic methods are used. The detailed and specific procedures for which are available in WHO guidelines as well as in pharmacopoeias. To summarize, we have considered the various methods for determining the quality of the materials. Hemolytic test and saponins. They are interrelated. Saponins are those materials which causes hemolysis and also have the properties of forming a foam. These two properties are utilized to determining the content of saponins in the plant material. Similarly, tannins are astringent materials are those materials which are phenolic in nature and they form with hide powder skin, and this is the basis of determining the total tannins in the material. Similarly, gums, mucilage and pectin substances swell, and the amount of material present in the herbal material which has the capacity of swelling are determined by swelling index. Microbial load is a major concern in most of the herbal material which are not properly collected, and therefore the limits of microbial load is very important. The Microbiological procedures for them are very detailed, and that is why we have not discussed the strain, the bacteria, and various other details which you can find in any standard textbook. Aflatoxin is another uh, area of great concern because a little quantity of aflatoxin can cause great toxicity, and that is why it is important to test them with a reference solution. Similarly, insecticide, fungicide, herbicide residues are also a matter of great concern because they also produce toxicity, and therefore their limits also are required to be measured when a material is really put to use. The detailed procedure of them are very lengthy, covering almost one lecture. We advise to refer to WHO guidelines and respective pharmacopoeias